it's a real privilege to talk to audiences such as the people we have here, because it keeps people like me on the right path, and the right path is to push forward to try and find out new ways of diagnosing these terrible tumours, and also, as it's just been mentioned, with the ultimate game of finding a cure. So I've been asked today to talk to you about the progress that we've managed to make with setting up the tumour bank. And as will become apparent later on in my talk, I only take a very small amount of credit for this. The credit goes to other people in this room, and I think you know who I am probably going to be talking about. So why do we need a tumour bank at all? And I just want to give you a bit of background to that, and then later on I'll try and illustrate how the existence of a tumour bank will help you in your quest. So basically, what is the challenge of a rare tumour? So when the people in here who have the tumour were diagnosed, no doubt their physician or their surgeon looked quite excited because this was a rare tumour, and it's very dull treating the same sort of cancer day in and day out. So having something unusual probably perked him or her up for a while, right? So you sort of say, oh look, they look excited, this must be good news. And so once that excitement wears away, they say, well, we must know exactly what to do then. A bit of a pause at that point. Now in some cases, of course, for your tumour types, he or she does know exactly what to do, but in other cases they don't. And that's partly because you have the affliction of having a tumour, but also a rare tumour. This means it's low down on people's priority lists when it comes to developing new treatments, and also knowledge amongst the people who are treating it is not always as good as it could be if you had a more common tumour. So the challenge of rare tumours, I'm afraid, doesn't extend just to you. Next slide, please. I like this. Oh no, it's not you though. Yes. Yeah. Don't worry. Don't worry. So, if you take a pie graph, a pie chart such as this one, summarising all the tumour types there are out there in the world, you'll see the common ones: lung, breast, colorectal, prostate and stomach. That's the same on the list as appear just about everywhere in the world. For various reasons, those are the common tumour types. Then you get into a smaller groups of liver, cervix, esophagus and bladder, and then you get other, right? So if you look at that chart, the most common sort of cancer to have is other, all right? So what about this other group? What do they consist of? And the fact is that these rare cancers are not so rare after all, if you lump them together. And this really good paper came out a couple of years ago highlighting this and summarising the evidence we knew from the whole of Europe about these tumour types. It's a mixed family of tumours, okay? So they defined rare as greater than, less than 6 in 100,000, all right? That means about 30,000, less than 30,000 people in the whole of Europe with a particular tumour type. There are 186 different flavours within the other group. So some of these tumour types are vanishingly rare, and the pause gist would be within that category, for example. The outcomes of this group as a whole tend to be worse than for the common tumours. That's a bit of a broad brush. Within that group there's some very, very curable tumours and some very, very stubborn ones. But overall they tend to do worse than the common tumours. And the reasons for that are partly because, as I said before, the level of knowledge amongst the people who look after you, but also the development of new treatments is difficult when you've only got 5 or 6 patients in the whole country to do clinical trials on. And these cancers are unusual in the sense they occur across all age ranges. So in the blue line we've got the incidence of cancer in the general population with the common tumours, and you can see that cancer generally is a disease of older age. But the rarer tumours cover all age ranges. So the paediatric tumours, the tumours in children, are also rare tumours. So the problems that you have in treating those cancers are almost always in this group. So you can see in the red line the incidence is pretty even across the whole of the age range. And I can see that looking around the room. All age ranges within this group are just about represented. So I've outlined the problem. What's the solution, you may ask? The solution must lie in more research. I would say that because I'm a researcher. It pays my mortgage. But actually I also think it's true. More research, the more you know about the enemy, the more you're likely to be able to defeat it. More collaboration. You can't do this in one, even a large centre. even a place like the Marsden, one of the most important hospitals in the whole of Europe for the treating of cancer, doesn't see enough people to be able to do research in these rare tumours. There has to be integration of your research across the United Kingdom, Europe, the States, wherever. And the rarer the tumour, the wider the net you need to cast. This in itself generates all sorts of different problems, as you can imagine, cultural differences, language problems. But one thing about coming to a meeting like this is as I look around the room, I can see a huge range of backgrounds and people. It brings together in a way that other things just do not do, those collaborations here. And you can see that replicated across different countries as well. It's something that brings people together. If you want to see people getting on best as human beings, this sort of environment is where you see it. So I'm very positive. Positive mental attitude, I agree with that, absolutely. Better surgery, so surgical techniques are important too. And just like for the medicines, that's a knowledge that needs to be shared between specialists. And more trials. Now, trials are difficult to set up anytime. Trials on rare tumours are doubly difficult. Trials on rare tumours you have to do between countries are triply difficult, which is another reason why it's quite difficult to get them off the ground. I think all of this, just about, is supported by the collection of samples for use in research, not only nationally, but internationally as well. And that because that supports a lot of the activity I've listed above, particularly the research side of stuff and also the trials. So getting samples into a biobank is a really important underlying infrastructure that we need to think about as a group. So what's the driving force? And we have a representative spectrum of driving force people here. And I'm looking at a whole load of driving forces. One of the really important things that has come out of the last few years in cancer research is the much better partnership between the experts and the people who have the tumour themselves, their interchange. I am looking at a room full of experts. I am terrified you're going to ask me the finer details of the mutation of the KIT molecule. I don't know it. So you are experts. I'm an expert in my own way. I'm an expert in the laboratory. I'm an expert in designing clinical trials to a certain extent. But you are experts too, and your driving force and your partnership is essential to make this moving forwards. So what progress has been made in this effort? We've managed to establish a loose consortium, and it's just a group of people precipitated by Jane's fantastic catalytic achievement who want to do the same thing, which is to want to generate this collection so it can be used in the ways that I've outlined. And we've collected, up to date, a massive total of 3. And I'm not being sarcastic, because actually it is a massive total in a short period of time in this rare tumour type. And through lots of meticulous planning by Jane and some sterling efforts, I think, by the people in the research laboratories, we've managed to get those samples collected, distributed to laboratories who are going to do research on the fresh material, and the sample has been collected and stored in our facility in Newcastle. The really important thing to note here is that I am not using those samples. I am looking after them on behalf of the researchers, the clinicians, and very importantly, for you. The other thing I need to point out is that we only take samples that are surplus to diagnostic requirements. That's the technical phrase. In other words, we only take material once the doctor who's looking after you or your loved one with this tumour has taken everything they need to be sure of the diagnosis. The other thing that always happens is that material is kept in the hospital where you were diagnosed and treated. There's always material there. We don't take it all. It would be wrong for us to take it all, because they may need to go back to that sample someday in the future. As we know in this tumour type, that could be 10 years, 15 years, and they want to look at it again to make sure that the diagnosis they made was absolutely what they would now call that tumour. Or they may want to look for something new, which wasn't apparent when you were diagnosed. They keep that material for at least 30 years, okay, even once you have died. So actually hospitals are chock-a-block of all these samples. It's quite a large storage problem for them. But just to reassure you that we do not take all the material. You'll have also seen Professor Wall showed some nice gruesome pictures of tumours. These are actually quite hefty jobs quite often, they're quite big. So most of that material actually gets thrown away. Okay? The pathologist takes the bit that they need. Hopefully they take a bit for us and research. There'll still be, remember that one in the stomach? Must be about that big. Okay? There's still plenty, and a lot of it will actually go in the bin because there's nowhere to store it. So it's unusual for these sorts of tumours to be short of material, but you've got to think ahead. You've got to ask for that collection to be made at the time of diagnosis. The other really important thing is that we cannot take that material unless you have given your consent. It's against the law. We can't go in and say, I've got a really clever idea and I'm sure it'd be fine if I took that tumour. They must realise it's a good idea. No, you have to give your specific consent. So a biobank is much more about having a big ring folder full of consents as it is about having a fridge full of samples. So getting those consent forms. So what if you've had your operation some time ago? Is it too late? No, that material will be still sitting in your pathology department and could be very useful for a research project. If we can get you to sign a consent form to say that should a project come up, can we approach the pathology department for ask for some of the tumour, then that would be immensely helpful. So we do have fresh material from patients, and that's the best. So if you know of somebody who's going to have an operation fairly soon, we would ask you to contact us so that we can get in touch with the hospital and get some material taken and put in the freezer. Because pathology departments don't store samples in freezers normally, they store them in paraffin. Freezers are very expensive and very expensive to run. Paraffin you can leave in a cupboard for years and it's fine. So that's one of the reasons they don't store frozen material routinely. Okay. What are the next steps? What we need to do is to form an access and steering group. And one of the things I've asked today of Barbara and her team is to think about how we do this. I'm not going to give that sample to anybody until I have permission to do so from a group of people who tell me that it's a good project to send it to. Otherwise, I might waste it on a useless project. So we need a group of people to make that difficult decision which says this project is good enough to do, but we don't wait for the perfect project, we wait forever. So having several people make that decision is really good. In fact, I wouldn't make that decision by myself because I'd hate the idea of you all getting very cross with me if I gave it away to something which wasn't worthy. So having that committee is really important. We need to get more samples. I've talked about getting more samples already, either ones sitting in the pathology department or sitting in our nice fridge in Newcastle. And we need to publicise the existence of this bank, and we've talked about how we might do that already today. If I've got a few seconds, can I tell you about one possible use for these sorts of samples? And it's something I call screen and recruit, which sounds a bit like wash and go, for those of you who've still got hair. And so this is an idea that we've come up in Newcastle and other places have come up with, which re-looks at the way that you do drug discovery. Right, this is the traditional way to do drug discovery. Take samples from people like yourselves. Look at them using molecular techniques in the laboratory. Find a drug target. Go away and design your drug. Then give it to the patient. Right? That's the normal way of doing it. And that's the sort of way it was done for imatinib and the way that it was done in chronic myeloid leukemia for imatinib as well. However, there is a different way. And the different way is to put the patient first. Take the sample first, screen it with your molecular techniques, and then go out to the drug companies saying, look, I've got all these lovely new molecular techniques. Would you like to go away and identify a target and then a drug? It's an important, different way round. It de-risks it for the drug company. Now, we often say lots of nasty things about drug companies, but actually, where would we be without them? We need them to develop the drugs. Okay? They need to make money. They don't have a heart. A drug company doesn't have a heart as such. It's not a person. It's an entity. It's something we've designed as humans. So therefore, we need to reduce their risk of losing their money by doing more of the science up front, by going to them with really attractive offers, saying, look at these wonderful targets in Paul's GIST. We found this. Go away and find me a drug. And that, I think, is the way round for rare tumours. So you screen first, recruit later. It's an upside-down way of doing it. You obtain consent of the patient at diagnosis to take the sample, but very importantly, you also say to the patient, if we come back with your sample, we found a target, is it okay if we give you a ring and see if you would take part in a trial? Now, believe it or not, ethics committees don't like you doing that unless you've got that permission up front. You wouldn't think that you would care, would you? If you had a sample and it had an exciting target, who could possibly object to you ringing up? Ethics committees don't like that. So we need people like you to sign a piece of paper saying, sure, if you find something that might be useful to me, please ring me up. If unfortunately I've died, my relatives will understand and not be upset, because that's what ethics committees are worried about, right? So that's something we may be putting into the consent form in the future. You obtain it, you screen it, and then you retain some of the DNA, the coding material from the sample, so that a new drug comes up. You can screen your collection very quickly and say, look, I found 50 patients here, it would be great on your new drug. Do you want to recruit them? And it's a way around it which makes it much cheaper for the drug companies to do their research. The other thing we need to think about is what I call mechanism-based trials. Okay, so normally, if you have cancer of the bowel, or if you had cancer of the prostate, you would have a trial related to prostate cancer or colon cancer, right? But if you look at it, those tumours at molecular level, they often have abnormalities in common. So you might find a mutation in the bowel cancer, which is the same as the mutation in the prostate cancer. Those are common tumours, so you don't have to worry about it. But in rare tumours, a way of getting the numbers up is to pool those people into one trial. So you're comparing somebody with a cancer, say, of the earlobe with a cancer of the left knee. What you need to do is decide what a good response looks like in those two different sites, which we should be able to do. Drug companies don't like it a lot, but it's a way of accelerating drug development in rare tumours. And some of the tumour types, the only way, I suspect, of doing it without just being anecdotal. In other words, treating somebody, writing it up, saying, wasn't that interesting? That's not a trial. So this is a way forward for them and for you to get new drugs more quickly through the trial process. More rapid patient recruitment, more rapid drug evaluation for the companies, saves them money. Reduced trial costs because you've done a lot of the work for them up front. Greater patient involvement, the patients are waiting for your phone call. It gives you that hope. And hope, I think, is vital, is it not? And this must not be confused with false expectation. Doctors are very worried about giving false expectation. They should never be worried about giving hope. There is a distinction in my mind. And the distinction is the conversation between you and your doctor is a mature, grown-up one, not a childish one. Okay? You know what they're saying. We can give you a bit of hope. We can't say with any certainty this will lead to a new trial for you, but it might do. And I'm sure people in this room are plenty grown up enough to be able to cope with that idea. So how are we going to make it happen? The patient register has been mentioned already a couple of times. Rapid mutation screening is something we could think about doing for your tumour type if we can get some money from various fundraisers for that. Not necessarily for you. I think this is the sort of thing we can get from people like Cancer Research UK. Public engagement and consultation and engagement of those pharmaceutical companies is the next steps. I've put this up and I hope it's not too brutal, but currently many patients with rare cancers will die waiting for that new trial. So we need to think of a different way of tackling this really challenging problem. And if we don't, I think we're letting everybody down. Thank you very much for your attention. I hope it hasn't been too brutal on the ears.